don't know me, I'm Mark Leitner. I have the privilege of being the CEO of the Alzheimer's Drug Discovery Foundation. I have a little bit of housekeeping, 2 notes of thanks, and an introduction. Anybody's keeping track. Just on the housekeeping, cell phones, if you guys put them on vibrate or silent or whatever it is we do with those these days, I'd appreciate it. We'll be going till about 11:30, and then for those staying for lunch, we'll break and then lunch and the surprise. 2 notes of thanks. One for a person who's not here today, our board overseer, Mr. Phil Lovett. He has underwritten us being here today, and we're very grateful to Phil and actually our entire board of overseers for what he does. The second note of thanks is to somebody who's just fabulous, and she's going to do her best not to hear anything I'm about to say, but I'm looking at her right now. That's Ms. Nancy Goods. Businesswoman, philanthropist, wife, fabulous. Nancy, thank you. Proud of you and your families here with us, which is just great. Back in April, Nancy and I had lunch, and she said to me, and every year, for those of you who are somewhat new to us today, was the annual Goods Prize. And that's given out to the top scientist that we feel is working on a clinical trial. And this year is the 5th anniversary. And Nancy said, I have an idea. And those of you who know and love Nancy, when she says, I have an idea, but listen, and for me, I take notes. And she said, I want to invite back all the past year's honorees and winners and bring them all together at one time so we can get a collective look at how that science is progressing. That's resulted in this morning, and with our wonderful title of What's Next After Apple. So thank you, Nancy, for your ideas, for your passion. I really appreciate it. Introduction. I get the opportunity to introduce someone I consider a friend and a partner, Dr. Howard Phil. He is a geriatrician, he is a neurologist. Those of us that know him most closely, he is a visionary. He was recently introduced out of the AAIC meeting in Los Angeles this summer by somebody from the pharmaceutical industry as the sage of novel therapeutics and repurposing approaches in Alzheimer's. Ladies and gentlemen, I give you Dr. Howard.
Thanks, Mark. And I want to thank all of you for coming today, which I hope will be a very exciting session. I think today that we're really presenting to you what represents a new era in Alzheimer's drug research. I know that for 15 or 20 years, we've spent literally tens of billions of dollars, maybe 30, 40, 50 billion dollars, both in academia and in the pharmaceutical industry on research on beta amyloid. And I know that all of you have read in the newspapers about our 100% failure rate since 2004. It is difficult to develop a drug. It's said that 90% of the people who work in the pharmaceutical industry will spend their entire careers never working on a drug that comes to market. And failure is the norm. But nevertheless, every study that we do in science, we learn something. And I remember my old mentor used to say that a good experiment was one that raises more questions than it answers. Today, we have the great privilege of hearing from 5 of ADDDF's funded investigators, all clinical scientists, in clinical trials with 5 very novel drugs for Alzheimer's disease. And some of them will present to you positive results today from the studies that we have funded. Many or most of the people that are before you today, we've been funding for 15 or 20 years. And it takes about 12 to 15 years to develop a drug and bring it to market. So that kind of time frame is not unusual in drug development. Our scientific strategy from the very beginning has been that aging is clearly the leading risk factor for Alzheimer's disease. And Alzheimer's is a disease of aging and old age. And the body of knowledge that underpins our scientific strategy really represents perhaps 100 years of gerontology research, of basic biology of aging research, and translating that research into new drugs for Alzheimer's disease. If you think about all the diseases of aging, heart disease, kidney disease, lung disease, they all have common frameworks, and Alzheimer's and the brain is also subject to these common pathways: inflammation, what we call epigenetics, genetic changes, neuroprotection, in other words, invoking the cell's normal responses to injury by promoting self-survival. And that some of the drugs that you're going to hear about today are going to be about how we're attacking Alzheimer's from an inflammatory point of view, from a neuroprotection point of view, from a hormone point of view, and also from the point of view of reducing oxidation in the brain. So ADDF, there are currently about 100 drugs in clinical trials for Alzheimer's disease. Remarkably, though we're a modest-sized foundation, a mid-sized foundation, we've supporting or have been supporting about 20% of all the drugs that are currently in clinical trials. About half of our drugs that we're supporting are in phase 2, which is the time that drugs are actually tested for whether or not they work. And you're going to hear about some phase 2 results today from some drugs that have actually gone through a phase 2 trial and shown very promising results that would justify going on to the next phase to see if they actually work. One of our niches to develop drugs, to test drugs for what we call repurposing. The best example of repurposing is a company called Celgene, which has become one of the most successful biotech companies in the world. They have a treatment for multiple myeloma. The name of that treatment is thalidomide. And most of you have probably heard about thalidomide. Thalidomide was a drug that was developed and used for pregnant women in the 1950s. It had horrible side effects and was taken off the market. But someone figured out that thalidomide might be useful for multiple myeloma. And sure enough, it worked. And so they developed a commercial strategy for repurposing thalidomide, testing it in multiple myeloma, and bringing it to market for multiple myeloma. One of the most successful drugs from the biotech industry. You're going to hear today about some repurposing experiments that we're doing. We currently have 19 clinical trials going on in which we're testing drugs that are indicated on the market, approved by the FDA. We know their safety profile, and we're using them to see if they'll work in Alzheimer's disease based on data from biology, based on data from efficacy that's been shown in animal models, and even based on data in human beings. We're also I can frame some of what we're doing also in the context of prevention. Some of our trials that we're funding, more than half of them, are in an entity called MCI, which is an acronym for mild cognitive impairment. So most of us think about Alzheimer's disease as a cause of dementia, which it is. But by the time people become demented, they're functionally impaired, and they have all kinds of deficits. And I know most of you have loved ones, and you've seen this up close and personal. But because Alzheimer's is a continuous disease that we know begins in middle age, begins in the 40s and 50s and 60s, and we can detect that through a brain scan that ADDF developed many years ago and brought to market as the first helped to bring to market. It was actually brought to market by Eli Lilly. But we funded that at Penn, as many of you know. And this brain scan really accelerated the way we're doing clinical trials. But it was the brain scan that showed how we can begin prevention for Alzheimer's by identifying the disease before there are clinical symptoms. So the disease, just like heart disease, it goes from a stage when there's only pathology, there's only the beginnings of the disease, through stages where people have mild memory loss and they don't have any functional impairment. If you met them at dinner, you wouldn't know that they had it. And then finally to dementia. And MCI is that stage where people just are showing the first clinical symptoms of Alzheimer's disease, but they're not functionally impaired, generally speaking. And so our sweet spot for clinical trials, for more than half of our clinical trials, is this MCI stage. And one of the practical goals of our clinical trials is to try to treat people with MCI to prevent them from becoming demented. I guess the last thing I want to say is that Our job as a philanthropy is to take risk. And we've been doing that from the very, very beginning. Take risk on novel ideas, novel targets, novel drugs. And part of what you're going to hear today from the panelists is really kind of high-risk experiments, high-risk drug development that we've been supporting as a philanthropy that investors wouldn't support because, you know, the for-profit potential is risky. And yet, you know, these are scientists that have spent their whole careers working on the drugs, many of them, that have been their passion for their whole, some of them for almost their whole professional life. And so I just want to applaud that these are 5 really novel drugs that are in clinical trials. Some of them have results, some of them are on the way to results, and all of them have really dedicated their entire professional lives, like myself, for almost 40 years now, some of them, to develop drugs for Alzheimer's. So I just want to impress upon you that this is and I say this humbly, a really impressive group of people that have all been recognized through the generosity of Nancy Goods and the Goods family as getting the Goods Prize, but they got the Goods Prize for a reason, and they're some of my favorite people. So I'm just very proud to be able to stand here and present this work to you today. Our first presentation of a drug is from Dr. Jeffrey Cummings, who I've known about 40 years, who's really, as we talk about clinical trials in Alzheimer's disease, I think it's very fair and very accurate to say that Jeff is probably the most recognized, most sought after consultant by industry and academia, the best clinical trialist in the world. And I say that without hyperbole. Jeff is the director of the Lou Ruvo Cleveland Clinic Center. He's a professor of neurology and other things at the Cleveland Clinic. I got to look in my, okay. And he's the director of the Center of Neurodegeneration and Translational Neuroscience at the Cleveland Clinic, professor of medicine there. He's the founder of that clinic in Las Vegas. If you ever happen to be in Las Vegas, I recommend that you visit the center that Jeff developed, because it was designed and built by Frank Gehry, who built the Bilbao and the Bilbao Museum, and it looks a little bit like it. It's really an impressive place. And Jeff is going to tell us about his recent clinical trial of a drug for Parkinson's disease that is the only drug, or one of the only drugs in Parkinson's disease that's ever been shown to slow down the course of Parkinson's disease. And he's got data and results from a clinical trial that he's going to tell you about that are positive results in a phase 2 study for Alzheimer's disease.
Thank you, Howard. Shall I stand up?
Yes.
Okay, great. Because it's hard for me to see you back there if I don't stand. Can you hear me? I noticed someone was sort of struggling there to hear. Can you hear me back there? Okay, great. So it's a delight to be here. And with a 40-year friendship, the biology of aging is speaking for itself here. That's a special part of this whole award. And thanks to Nancy. Where's Nancy? There, you moved back there. So thanks to Nancy for making the Goods Prize available. It's great to have your work recognized, and these are the people that I turn to for guidance and advice and inspiration, and it's fantastic to join them as a Goods Prize winner. So we wanted to test a drug, rasagiline, to see if it would be beneficial in Alzheimer's disease. But as Howard said, every trial shouldn't be just about the drug. It should be about the process of the trial and the process of drug development. So then the question was, could we accelerate drug development? And within that, could we accelerate a clinical trial? So rasagiline was chosen because we looked at the Parkinson's data, and yes, it improved the motor symptoms, but on the different scales that were used to measure cognition, there was the cognitive signal showing through. So I thought, well, that's a really interesting observation. Then we looked at the basic science literature And this drug has a plethora of effects that address some of the main issues of the biology of aging. So we thought, well, this is sort of starting to come together to tell a story. Then we looked at other drugs in the same class. One particular called selegiline was tested in 1997 and shown to slow the progress of Alzheimer's disease, and it was never followed up. So we said, well, this is enough for us to begin thinking about a clinical trial for Alzheimer's disease. It was, how would we accelerate drug development? Just as Howard said, through repurposing. The timeline for a repurposed drug is about 40% shorter than the timeline for a drug that starts off as a white powder in a laboratory and has to be tested in animals. So you're really accelerating the drug development program if you can make this work. How do you accelerate the trial? In our case, we made the readout a biomarker. It takes a lot of patience and a long time to show that you've actually changed human cognition, because unfortunately we all have good days and bad days, and that creates a lot of variability in the data. But your brain metabolism is quite stable, and therefore if I could show an effect on brain metabolism, I could do it in a smaller number of patients. Moreover, if I didn't show any effect on brain metabolism, then I would conclude that that drug should not be advanced. And we need more aggressive in terms of stopping drugs that have no hope of success. So that was the way we decided to shorten the trial, was the use of biomarkers. We used two of them, brain metabolism, and the other, which Howard made available to us, was a measure of the neurofibrillary tangle. A brand new kind of scan, not even approved by the FDA yet, so still an experimental scan, but tells us whether one of the key pathologies of Alzheimer's disease has been delayed. So we have now completed the trial. We did it in the Cleveland Clinic system. We recruited only 50 patients, so this is a very small trial, but we had powered it around the effect on brain metabolism. There was no question that we had a statistically significant benefit in the treatment group compared to the placebo group. A scientist hardly ever gets to say that. So it was really great. It's a positive outcome. It's a positive trial. And it's the kind of positive trial that says we had an effect on the biomarker, and we should now do the kind of trial that would tell us whether it had a clinical benefit. Now, did it have a clinical benefit? What we saw was that in all cases except one of our clinical measures, the drug outperformed placebo numerically. We did not have enough patients in the trial to show that it was statistically significantly greater, but it was very consistently better. And in the case of a quality of life measure, it was statistically significantly better. Moreover, the most provocative observation, thanks to Howard for making this happen, was that the tau signal was also reduced in the treatment group compared to the placebo group. So the conclusion we would most like to draw is that we slowed down the formation of neurofibrillary tangles. However, we're a little worried that we don't know enough about the chemistry of this molecule to be certain of that outcome. So we are not concluding that based on this trial, but we do have a strong reason to look at this and other related biomarkers in other trials. So we are extremely delighted with this result. We have now submitted it to the federal framework for clinical trial testing, which would be the next step to see whether we can eventually make it available to people to help their lives if they're suffering from moderate Alzheimer's disease. What did ADDF do? Well, ADDF provided funding. That's fantastic, right? We couldn't have done it without it. But maybe just as important, they directed us to the scientist who knows exactly how to do the metabolic brain scanning. So we joined with her. This is Dawn Matthews of ADM Diagnosis. She's a terrific scientist, and she leads all the analysis of our metabolic brain imaging. She also, because of her expertise in brain imaging, is doing all of the analyses on the tau imaging. So that relationship has been extremely important, and Howard and ADF were able to connect us with Lilly for the tau imaging, with Dawn for the FDG imaging, and provide us with funding. So we just couldn't have done this. I couldn't be standing here with my enthusiasm showing without the support of the ADDF. So we will now take this drug to the next step, and it's going to be greatly facilitated by the Goods Prize because we didn't anticipate the tau effect. So we have new analyses to do. We have blood on these patients, so we're going to do new biomarkers. We're going to plan this trial that will show whether there's clinical benefit, and all of those things are going to be accomplished because of the Goods Prize. So we are absolutely delighted that this has all come together to allow us to take this to the next step so that we can eventually deliver a drug to the patients who need it. Thank you.
Thanks, Jeff. Thanks, Jeff. And just to be clear, because maybe I didn't say this, and you probably all know this, but Jeff is this year's honoree for the Goods Prize. So that's why he's able to tell us exactly what he's going to do with the new. Our next speaker is Marty Watterson, Dr. Marty Watterson. And Marty is what I would call a drug discoverer. Who's actually in the aeronautics industry originally, has a wonderful career. But he's actually at an academic center where some of the most successful drugs in history were developed. And many of you may have heard of Lyrica. Lyrica was developed at Marty's center and is an example of how, and actually a very early example of how drugs can be discovered and developed at academic centers. Marty doing this at Northwestern. When we think about basic research, let me say that we must think about biology. That's where we learn about how disease processes work. But when we think about developing drugs, this is much more about chemistry. And this is one of Marty's strengths. Marty and his group are a group of really sophisticated chemists. They can look at a biological target and then think about how, you got this molecule which you can't see except you kind of just have it in your mind's eye or on a slide somewhere, or somehow you figured out this structure. It's just incredible. I mean, these people are wizards. These medicinal chemists. They're chemists that create medicines. They're chemists that synthesize. They conceive based on the structure of a molecule that none of us will ever see, and they can see the structure. And they can then design a molecule through chemistry that fits into that structure and does the right thing and then becomes that little white pill that you take by mouth and it cures a disease. It's just amazing. And that's kind of what, in a nutshell, Marty does. He's the John D. Searle Professor of Molecular Biology and Biochemistry at Northwestern, and he's a founder of a new biotech company that was spun out of Northwestern called Neurokine. The focus of their drug is inflammation, and not just any kind of inflammation, but very specifically brain inflammation. We've had a lot of research going back almost 40 years, actually, because when I started at Rockefeller 40 years ago, we were interested in inflammation. But that idea that there's inflammation that plays a role in the process of Alzheimer's disease back then was completely discounted. And then in the 1990s, there was some work on testing drugs like Motrin for Alzheimer's disease and Vioxx and Celebrex, and they all failed. Maybe they were anti-inflammatories, but they were the wrong anti-inflammatories. So what we need is anti-inflammatories that what I would call are neuro-anti-inflammatories that specifically affect the cells in the brain that are responsible for the inflammation that many of us now think are the key process that even though many of us may have amyloid in our brains, some of us don't have a really robust inflammatory reaction, but those of us who do are the ones who develop symptoms and progression of the illness into dementia. And so there's a lot of interest, and actually the largest part of our portfolio right now is about neuro-anti-inflammatories. And Marty has been, again, working for many, many years on developing this neuro-anti-inflammatory that he's going to be talking to you about. He recently spun out a company that ADDF has made an investment in, and they're in phase 1 clinical trials now in Europe, where they're testing the safety of the drug. And we're very excited that we believe this drug will be the first in its class that's shown safety, and then we'll go into investing in the phase 2 study to show whether or not the drug works. So let me hand it over to Marty, who, again, everybody on the podium here has gotten the good ride, so we're very proud to be able to do that.
Thank you, Howard. So picking up on your theme of the biology of aging and things that happen is, I guess you would call them comorbidities, that happens across several diseases also. So as far as the inflammation, covers a wide area. So the drugs that were mentioned are ones that affect lipids that are made in your body. So we focused on ones that affect certain small proteins that clinicians have previously shown are linked to what's called pathophysiology progression, disease progression. And it is definitely implicated in animal models and in clinical samples for Alzheimer's, for other neurological disorders such as certain types of autism spectrum disorder, and certain types of other progressive diseases. So these are all complex diseases. So the first thing we wanted to do, besides actually going after this target, which a lot of people had tried to develop a drug for but had failed mainly because of safety, was to make something very safe. So it was safety first. And the idea there was you would want to be able to take it every day for the rest of your life, starting at a certain age. So safety was a high priority. Also, when you have something very safe, the probability of combining it with other drugs in the clinic is greatly enhanced because off-target or side effects of two drugs is not 2 + 2 = 4. It's usually much worse than that. So there's no way to forecast that. So this allows new drugs to be combined with existing drugs. So that was our basic approach. So in this particular drug, which is a recent spin out of the academic work, the phase 1A has gone very well, placebo-controlled, double-blind in Europe, and it's remarkably safe. Again, we had no idea which was which. We knew it was going well because there were no reports of adverse events. Phase 1B, which has to do with accelerating even more, is also going along quite well. This work would not have been possible without ADDF's involvement. In fact, I would say regular NIH funding mechanisms as recent as 5 or 6 years ago looked at this askance. Some people even said because we weren't directly attacking amyloid, because in fact we found out this drug and animal models did not affect amyloid pathway, we were told it was going to fail. It would never progress. So in animal models it's worked well. In humans it's very safe. And we also, in parallel, because this is a new drug, the good news and bad news about novel compounds, new chemical entities, is its innovation. If it's first in class, it's innovation. From a regulatory perspective in patients, that's double risk. So the safety is important. So the ADDF also recently funded what are called extended toxicology trials in animals. If you want to do an Alzheimer's trial in which you're going past 1 month or 6 months, you want to go for longer periods of time, you have to have this certain goal be achieved in your preclinical work. So those are being finished now and very promising. So at this point, we are actually planning phase 2, first in patient trials, and discussing with experts who are in the clinical trials about how to structure this in terms of biomarkers, which we're working out the assays for now. The ADDF, by co-sponsoring that work along with the clinical trials and the safety, cut about 2 years off the process. So normally, these extreme safety things, you're going to be have a timeline that spreads out. So I would call ADDF not only an innovation funder, but a catalyst of the progression. And the timing was perfect when I received the Goods Award because we were at the process of doing our favorite thing as scientists, and that's paperwork and filing for regulatory approvals to start. So And that costs money. So that funding was actually used by the spin-out company to do a fast-track safety phase 1AB approval together, which it did, and that put us another year ahead. So we're actually in 2020 looking to set up first-in-patient trials, which, considering the fact that the patent was issued in 2017, our trial paperwork started in 2018, our trial started in the beginning of 2019. This is quite remarkable, and it's not so much us, it's actually those who have the foresight to catalyze and sponsor this type of innovation, and something that is different from what the mainstream is doing, but it's something that is just as justified scientifically and medically, but there has not been a tax on it because of the risk. It's risk to the careers of the scientists, it's risk to the funding agencies, but clearly this is where we're going to have to go in terms of coming up with alternatives to the learning curve we have from plaque-based failures. Thank you.
Thank you, Marty. So our next scientist is Dr. Roberta Brenton. Robbie and I have also known each other 40 years. I had the privilege of introducing Robbie at Rockefeller University when I started the Alzheimer's clinic there in 1981 to her first Alzheimer patient, and Robbie will tell you, I'm sure, that how it just meeting a patient, she was a bench scientist, meeting a patient changed her career. Robbie is the inaugural director of the Center for Innovation in Brain Science at the University of Arizona, where she also serves as a professor of pharmacology and neurology in the College of Medicine in Tucson. And she has spent her entire career around a hormone that's related to estrogen called allopregnanolone that goes down with aging and has tremendous metabolic effects that she'll tell you about. The drug itself was recently approved for use by a biotech company for postpartum depression. And her innovation is just to use this drug in Alzheimer's, which is something she's been working on for a very long time. And we're very excited that this drug will really have a neuroprotective and metabolic effect in Alzheimer's patients. And so looking forward to hearing Robbie
Thank you,
talk about
Howard.
it.
Well, it's wonderful to be back in New York, where this story, as Howard said, began. And it began with a single person with Alzheimer's disease. She had Alzheimer's disease, and I would spend hours with her walking along the path of the Rockefeller Children's Garden, now known as Rockefeller University. And she would tell me these wonderful stories about her career. And one evening, I spent several hours with her, took her back to her hospital room in Rockefeller Hospital, and I bid her goodnight. And I closed the door, and I waited 30 seconds. I knocked on the door and entered, and I said, "Dr. Ansbacher, do you remember me?" And she was so lovely. She said, "I'm so sorry, should I?" She could not remember me for 30 seconds, and I've remembered her for 30 years. And that opportunity to meet a woman with Alzheimer's disease changed the course of this woman neuroscientist. And when I set up my own laboratory in Los Angeles at USC, I began to try to understand what is it about that aging brain that puts that brain at risk for Alzheimer's disease. And in the course of that discovery, the course of that science, I made a remarkable discovery. And that remarkable discovery was that this molecule, allopregnanolone, created new nerve cells. Now, this was in a dish, but it was quite surprising. I came in back in the morning and I looked through the microscope, and there were more neurons than there were last night. So we then embarked on a program of research that was funded early on by ADDF and their innovative leadership that when I called and said, you know, it looks like we have the potential to regenerate the degenerated Alzheimer's brain. And instead of hanging up, saying you're delusional, Howard said, cut to the chase, Robbie. Do the experiment. Will it work? And we've been doing that experiment ever since. So thanks to the support of the Alzheimer's Drug Discovery Foundation and their early investment in this innovative and out-of-the-box idea, we were able to ultimately conduct a phase 1 safety trial with a twist. We were able to include in this trial some early indicators that might suggest that we had a beneficial effect. So the good news is it's very safe. The other encouraging news is that there are hints. There's a suggestion that indeed people with Alzheimer's disease can regenerate their brain. Now we're also now moving forward with a phase 2 clinical trial that was recently funded by the National Institute on Aging, built on ADDF support and the support from Nancy and Melvin Good's Prize. And in that trial, we're going to move forward using and developing a precision medicine approach. It appears that allo, A-L-L-O, is most beneficial in those who carry the risk factor gene for Alzheimer's disease, the ApoE4 gene. So we're moving forward for a precision medicine approach to our therapeutic development of allo for Alzheimer's disease in this high-risk population. The Goods Award allows us to advance our therapeutic development into something that has been talked about before, the patentable range. The ability now to take our natural molecule. Let me ask, how many women in the room have given birth? You made a lot of this molecule. We all make this molecule, but in the 3rd trimester of pregnancy, women make a great deal of that molecule, allopregnanolone. It's a daughter of progesterone. And because that there have been thousands of years of safety testing in women of reproductive age with fetus, that safety testing has already been done by all of us for our entire lives. That molecule is not patentable, but thanks to the Goods Prize, we will be able to patent formulations of that molecule, the ability to administer this molecule through the skin or under the tongue. Our initial trial was through intravenous infusion. Our next trial, our bridging studies, being conducted with intramuscular injection, with allopregnanolone from support from the ADDF. So in totality, we have the potential to bring a regenerated brain, these neural stem cells, to regenerate those neural stem cells in the brain so that individuals with Alzheimer's can regenerate their own brain. And thanks to ADDF and the Goods Prize, we anticipate bringing that remarkable discovery to life. Thank you.
Thanks, Robbie. Robbie is a really good example of how early support from ADDF over a period of many years ultimately led, and Robbie was rather modest, but she just got $37.5 million from the government to fund her phase 2 trial. So congratulations.
Thank you.
Another similar story is Dr. Makayla Gallagher, who's been a neuroscientist. She's the head of the Neurogenetics and Behavior Center at Johns Hopkins, and she's the founder of a biotech company called Aging Bio. And her research holds the potential to also slow the progression of symptoms for Alzheimer's disease, particularly, as I mentioned earlier, in patients with mild cognitive impairment. And we've been supporting Makayla for a number of years, and
You
look
bet.
forward to hearing from her.
Thanks a lot. So I've also been a faculty member, researcher studying the biology of aging for nearly 40 years, and now I'm doing it. So, and I'm on a mission. And I'm on a mission as a scientist at Johns Hopkins and at the company that I founded with a tremendous team. Our company is now doing a phase 3 trial. So this is a trial that could lead to the registration of a drug that can be prescribed. It is repositioned, so it is the safety database. The FDA has no concern about that. The use of this drug in Alzheimer's disease, which started in my work at Johns Hopkins in 2009-10, is now even more strongly supported by our current thinking post-amyloid about a direction we should be going. So we discovered that there is overactivity that occurs in the brain situated in very specialized circuits for memory. That occurs in aging and augments in mild cognitive impairment as people get into this transitional phase. Now you might think, and people at the time thought, as they were measuring this with brain imaging, the activity in the brain and its localization, that that might be working harder to make memories. But it turns out the memory system, memories get degraded when it's overactive. So you get very noisy memories. And then overactivity can lead to a condition that is pathological, even for healthy neurons. And now, today, what we understand is that neural activity is driving the spread of pathology from one neuron to another along connectional pathways. So now you will hear a lot in the future about therapeutics that are directed at tau in tangles. Tau is initially confined in a tiny area in the medial temporal lobe in the lateral entorhinal cortex. And over the course of mild cognitive impairment, this transitional condition, it begins to spread from neurons that are there to neurons that it would normally be talking to, and high activity augments that spread. And so we found a drug that we could, in animals, and then showed it in humans using this imaging, normalize that overactivity precisely in those locations. So what we are doing in our phase 3 trial is putting patients with mild cognitive impairment on this drug for a longer period of time to see if we can halt their progression and prevent dementia, because they still are quite functionally intact. If you can prolong that transition, which is at least a decade in mild cognitive impairment, for 5 years, you decrease the prevalence, the number of people with dementia, by 50%. That would be great. But that's not the end of my mission. My mission is to stop it entirely, to put it in the history books. We have now begun to learn, and this is also a separate project funded by ADDF for biomarkers, what the mechanism is that is causing that overactivity in the brain. And there is a key substance that you can measure in the cerebral spinal fluid, and people I collaborate with Hopkins are getting it in blood, okay? And this substance tracks whether you have a mechanism, the brakes in the brain, to slow that overactivity. And as your brakes are failing, this substance is going down. You can measure it in cerebral spinal fluid. The amazing thing, and I was at a meeting last week at NIH, is that in asymptomatic Alzheimer's disease, these are people maybe age 90, autopsy, the pathologist says Alzheimer's. Everyone who knew this person says no. They were clinically normal. And in this brain material, this substance is maintained. So it could be that you absolutely need the overactivity as an additional hit, along with the pathology, to make this whole thing neurodegenerative in the first place. So what we have gotten from the Alzheimer's, I can't begin to tell you, they've done everything for us. Right now, the Goods Prize that I was awarded last year, we're learning how to analyze, because in our phase 3 trial, we're using these tau imaging agents so we can see the tau spreading in the brain in the people that are enrolled in the trial at baseline and what happens over the course of treatment with the drug. If the theory is correct, this treatment with this drug should keep tau from spreading. As long as we keep it there, you're not going to get the traveling and the degeneration in the brain that is manifest in this protracted, expanding, awful deterioration of thinking beyond memory itself. So I'm very excited about that. We want the biomarker because if this works, then we can do the biomarker in people in their 40s and 50s. And if you start seeing the substance dropping, it's like cholesterol. Take your statins. We'll take a drug to basically treat, and that's called a companion diagnostic. Amyloid is a biomarker for Alzheimer's. If you've got it in your brain, something's wrong. But it doesn't, you can remove the amyloid and it doesn't change the disease. This is a companion diagnostic. It's like cholesterol, that if your cholesterol is going up, take the statins. If this biomarker is going down, it would say that you have a problem in the regulation of activity in your brain that can be contributing in a key fashion to the emergence of the disease in the first place. So that's my dream. I'm not going to stop working, till we do that. Thank you.
I just want to point out 3 things about Makayla's work. First is Makayla spent her entire career in academia at Johns Hopkins as a very renowned scientist, but she's become a biotech entrepreneur, okay? And these are the kind of people we need. She's now basically the acting CEO of this biotech company. She's out there. She spent the last couple of years learning how to be how to develop drugs in the biotech sector, and she's leading this biotech company forward. The second thing is that her drug is actually an example of repurposing. And one of the problems with repurposing is how do you create commercial potential out of a drug that's already on the market for other indications. And I won't go into the details of it, but basically her method is taking advantage of the fact that the dose that she's using is different from the dose that's approved on the market. So from that she can get intellectual property and move forward to get this drug to the market. And the last thing I want to point out and emphasize is that, to my knowledge, her drug is the only drug that's currently in phase 3 for Alzheimer's, the phase before approval, that's not an anti-amyloid approach. And last but certainly not least is Dr. Frank Longo. Frank and I started working together in 2002, I think it was, when Frank was at UCSF. And he had an idea, a crazy idea, if you will, that you could take a molecule called NGF, which had been tried for Alzheimer's disease in the past. It's a big hormone. It's one of the most neuroprotective hormones, I'm sure Frank's going to tell you what that means, that we know about in the brain. And he wanted to make a pill out of it because you can't drug NGF. It's a big molecule. You can't take it by mouth. It had bad side effects because of complex biology. And Frank actually started his work on a computer using a database of 2 million compounds. And knowing again the structure, as I pointed out, he had what we call a computational scientist who managed this thing, and 2 million compounds took almost a year. Today you can do it in about a week or 2. He found 60 of those compounds that actually bound in the proper way. He found 2 of those that he bought. Today, fast forward, we followed Frank to the University of North Carolina, where he chaired neurology there. We were founding investors in his biotech company, Pharmatrophics, around 2007, I think it was. Right. And we continued to fund Frank through that period. He moved to Stanford, where he's now the chairman of neurology and neuroscience. I don't know how he does it. He sees patients, he runs the biotech. Well, he doesn't run the biotech, but he's very involved in the biotech, and just a Renaissance man. And now Frank, after these, 17 years of research, is in phase 2, and phase 2, again, looking at efficacy. This drug, if it works, I promise you, and I don't use this word very often, and I'm not prone to hyperbole, it will be revolutionary because it'll be the first neuroprotective agent for neurodegenerative diseases.
Thank you, Howard. So thank you, Howard. And yeah, I've gone to every scientist on this panel for advice over the years, so it's a privilege to be up here with you guys. So what happens in the brain during Alzheimer's disease? And basically the bottom line why patients come to our clinic at Stanford or anywhere else is we're losing the synaptic connections, right? The neurons in our brain communicate with each other through these connections we call synapses. And over the years, we're losing the function of those synapses, and then we finally start losing the connections themselves. So the bottom line is to prevent or reverse that loss of communication between those synapses and reverse the loss of those synapses themselves. There are many powerful forces promoting that. We don't understand them entirely yet, but 4 big ones are probably the accumulation of amyloid in the brain. Most of us think that amyloid is still important in spite of the failures recently targeting that, but the problem is there are probably additional forces, right? So we have amyloid, we have the second protein my colleagues mentioned, the tau protein, toxic forms of it are accumulating. The amyloid damages the synapses, the toxic forms of tau damage the synapses. A third powerful force is probably, Dr. Watterson mentioned it, neuroinflammation. Toxic forms of neuroinflammation in the brain. And finally, as Howard mentioned, age-related degeneration itself, just getting older. So there are 4 powerful forces, and one might think offhand, wow, how do we develop a therapy counteracting even one of these powerful forces, let alone all 4 and however many more there are, right? So as we thought about this, and as the research we were doing, we recognized that what's common from all of these forces is that in some combination, alone or together, they trigger what we call a degenerative signaling inside the neurons. So each cell in our body has chemical signaling processes that tell that cell what to do. For example, these chemical signaling can tell a cell, don't divide or divide. And if you're inappropriately signaling to divide when you're not supposed to, you've got cancer, right? Well, similarly, in the nerve cells, this signaling tells you, keep that synaptic connection, it's important, or don't keep it, let it degenerate, right? So in Alzheimer's disease and with aging, the chemical signaling in the neurons is telling those synapses to degenerate. So we recognize that on the surface of the neurons, there are these receptors that control this degenerative signaling. And they have a normal job. And we thought if we could target these receptors with therapeutics, many of the drugs that we use today target these proteins on cell surfaces called receptors that are powerful ways of telling a cell what to do. So there were 2 receptors that we were excited about. One's called the P75 receptor and one's called the TrkB receptor. And we thought if we could target those receptors, we could counteract these degenerative processes, whether they're caused by amyloid or inflammation or tau, or even aging. The problem was there were no one had ever tried to target these receptors before. We were starting from scratch. There was nothing, no drugs, no one had tried it. In fact, I was told by a number of experts this was not even possible. I was fortunate to have had NIH grants, but my colleagues at NIH said, Frank, this sounds interesting. When you have some preliminary data, put in a grant. So I was going nowhere fast. And then I got one of the best phone calls in my life. I say the best was one from my wife early on, but one of the best was from Howard Phillip, who said, Frank, tell me about what you're trying to do. Anyway, ADDF got us off the ground. We exist today because of ADDF, and we started on this process. To make a long story short, we successfully invented drugs that target that first receptor, and now with help from the goods funding, the second receptor. As Howard mentioned, our drugs targeting the first receptor made it through 2 phase 1 studies. Phase 1 is so-called normal humans. When you're inventing the first drug in an entire class, safety is important. The FDA requires testing in normal people. That was a challenging experience. I was there for the first human taking our drug. It was kind of scary because you're worried about safety. And fortunately, that first human who volunteered to take our drug had no side effects. And I asked him afterwards, why did you risk yourself taking a drug that only a mouse has taken before? And he said to me, Dr. Longo, it was the least I could do. I was brought up by my grandfather, and he died of Alzheimer's. So you see the real courage that goes along with this. Our second drug group that the goods funding is provided targets the second receptor, and we've made a recent discovery with it that I wasn't sure was possible. You know, we hear so much about every time we have one of these big phase 3 trial failures going after amyloid, what we hear people say is, well, we treated the disease too late. We've got to start early, and they keep moving it early and early. We have to start treating people before there are any symptoms, way early. It's hopeless to treat people late. I disagree with that entirely. I'm not going to give up on the 50 million people in the world that have a diagnosis of Alzheimer's. I believe the biology that we're harnessing is powerful enough to reverse or even promote regeneration. And so this past summer, we reported in Los Angeles at the Alzheimer meeting, we could take brain slices from a very late stage mouse, and we tested, we used these sophisticated electrical ways of looking at that synaptic function that we're losing in Alzheimer's. And the synaptic function is gone, it's lost by this electrical testing, right? So the experts would say too late. We added one of these drugs from our second category that the Goods Ward has helped us develop. And this was a long shot, I thought. We added that, and we were able to normalize the synaptic function. So the basic structure is still there for a long time in our patients, and we can. This tells me we can create therapies that can get that synaptic function back, at least to a significant extent. And our patients give us glimpses of that. You know, I follow patients at Stanford who unfortunately are pretty advanced, but then their family tell me, wow, he had a really good couple hours. It was amazing. What is that telling us? The basic synaptic structure, a lot of it's still there. Let's not give up on that, right? So I think these drugs have been very powerful. And the Goods Award has also allowed us to do something that's really important that no one would ever, people would think is not scientifically exciting to fund. So to get these drugs through phase 3, we'll partner with a large pharma at some point, or it'll take a significant investment. And no one will make that investment unless the intellectual property is strong with this molecule, right? A pharma partner is not going to invest $300 million with weak intellectual property. So we're lucky we have one of the top firms in Silicon Valley, Wilson Sonsini, managing our intellectual property. And they send us lawyer bills every month. And so, you know, as a scientist, it's not very interesting to me, but if I want to get these drugs to people, we've got to have that intellectual property. So we've been able to fund that too. And now we have a really robust intellectual property portfolio managed by Wilson Sonsini. So that's been great too. It's just one of the ways, you know, we want to get drugs to people. So, you know, thank you for your support. It's been great.
And again, I want to point out Frank, chairman of neurology at Stanford, but also a bio-entrepreneur to the degree that he's founded this company and has helped to manage it through. And it's the only way that we're going to get drugs to market, as he mentioned, and IP is so important. I want to open it up now to questions from the audience. If anybody has any questions, we welcome any of them. Yes, Buzz?
I was always curious, is there a large database that includes all of your work that other scientists can draw on?
So I'll repeat the question. The question is there a large database of all of our work that other scientists can draw on? We're having trouble hearing back there. As a nonprofit, all of the work that basically, I won't say all the work, but the programs that we fund are all on our website, okay? And if any of you are interested in learning more about the programs, obviously their work, and especially because we're in this field of drug development, intellectual property is key. If you don't have intellectual property, if we don't protect their data, then there's not going to be a drug, period. And, you know, no pharma is going to invest, as Frank said, $1 billion, not 300 million, but $1 billion to $2 billion in a drug where there's no intellectual property. So we have to protect their private rights, okay? But if any of you are interested in investing in Makayla's company or Frank's company or Marty's company or any of their programs, let me know, because then we can put you in touch with them. If they're willing to share under a confidentiality agreement, then that can happen, and we'd be happy to connect you with that. So the starting point is either what you hear, or go to our website. You'll see all the programs. They're briefly described, and if you have more interest, please contact me. Chuck.
Howard, can I respond to that
Yes,
as
please.
well? I think this is such an important question because it's the data sharing that allows us to advance the field. One mechanism of data sharing is that every clinical trial conducted in the United States is required by law to be registered on clinicaltrials.gov. And now, since in the last 2 years, you're required by law to put the results on within 1 year of completing the trial. Now, it's like the post office running a clinical trial, so these data are not so easily accessible that you really find this a very workable website where you can put all the data together easily. But the data are there, and it's required that they be there. So there are some mechanisms in which we can get to these kinds of results.
Yeah, thank you, Jeff. It's a really important point that Jeff makes. I just want to point out, like, we talk a lot about data sharing, and when it's basic science and digital data and so on, that's easy to share. But again, because of the IP issues, like for example, there's publication issues in academia, but people should delay their publication until there's intellectual property on the data so that it can be protected. But as Jeff mentioned, and I appreciate the point, if it's a clinical trial, then the data have to be reported. Yes, in the back.
You mentioned,
Could you speak up if you can?
yes, thank you all very much. You mentioned 3 kinds of capital: philanthropic capital, investment capital, and governmental capital.
Yes.
Can you talk about the utility of each, the access to each, the forecast for each?
So the question is about capital from philanthropy, investors, and government. Let me start off with investors. We've had 100% failure rate in drug development for Alzheimer's disease since 2004. It's about, I think, is it right, 450 trials? Mm-hmm. We've had 450 basically failed trials on about 250 drugs, okay? And now the average venture capital fund is a 10-year fund, and it takes 12 to 15 years, and in some cases, you see, we've been supporting programs for 17 years, and they're in phase 2. So, a venture capital group or an angel group that's responsible to their limited partners to be responsible investors and make a profit are just not going to be able to invest in this thing, okay? So that's one barrier to for-profit investors, private investors. There's a couple of funds out there that are early-stage funders that will take risk, but that risk is a very small part of a venture capital. Government has come a long way out. Yes, sir.
Maybe just staying on the investment capital, can you move from VC all the way to big pharma, which has reduced its R&D and therefore it's buying its pipeline, if you will? And you mentioned protected data, so the coordination with for-profit capital, but corporate as opposed to VC, which can pull things along.
Yeah, so we work with and dialogue with corporate pharma venture funds. And we've interacted a lot with Pfizer and all the other big pharma. As many of you know, because there's been 100% failure rate in our field, and in CNS, central nervous system, and psychiatry in general, most of the big pharma have exited internal drug research and development for psychiatric and central nervous system neurodegenerative diseases. Just doesn't work for the public markets. What they've done is shift investment to external R&D, so you've got a lot of scouts out there in academia and so on, looking at opportunities. But the number of investments that big pharma is making through their venture funds in neurodegenerative disease is very limited. It's picking up a little bit. I think the role of big pharma has changed. They've closed their internal R&Ds, big buildings, brick and mortar type approaches, and they're waiting for things to percolate, okay? So basically, get a biotech like let's say Frank or Makayla or Marty, and once they get phase 2 results and they're impressive results, big pharma is going to be all over this, and probably venture capital. But as Frank just told you, it took 17 years of philanthropic and government funding to get to the phase 2. So big pharma has a role. They're the only the only people with $1.2 to $3 billion that can do the phase 3 and bring drugs to market. But their role has really been further down the line. Government has taken. Go ahead.
Oh, I'm sorry. I wanted to say something about that.
Yeah, please.
Because, Robbie, the government funding, because of the bypass budget, has really been terrific. And the company that I started did a Series A. But since the Series A, the company has secured $45 million in NIH funding across 2 programs. And, I think that's smart money. The issue that we are having right now that ties into everything you've been saying about failures is that because we are doing a repositioned pill, and we have gorgeous IP all over the globe for that pill, that still is not necessarily a big pharma model. But CNS Pharma is interested in, because we're not going to have the sales force, to sell the pill when the trial is successful. So you still need the tie into corporate.
So I'd like to take this opportunity, which is really fantastic, to announce our biotech company, New Therapeutics. It's NEU Therapeutics for neural therapeutics. So this is a very thrilling moment for me to actually announce for the first time to you, Howard, and to your audience, New Therapeutics.
Thank you, Robbie. And I just want to say the NIH budget has grown from, for Alzheimer's disease, has grown from $500 million a year. It was one of the smallest institutes at the end. The National Institute on Aging, which funds the Alzheimer's Research, was one of the smallest institutes, the lowest budget about 5 years ago, and for all the prior 40 years, to about the 4th or 5th largest institute now with about $2.4 billion. And the government has also shifted from an ivory tower, basic science predominance in research to more of a drug discovery and development engine. And as was mentioned, they're starting to fund drug development. So government is playing an important role. The value of philanthropy is in taking risk. It's in being innovative. It's being able to be quick to market. It takes about 18 months for an investigator to get an approval for a grant, a government grant. We turn around our projects. We try to turn around our projects in 90 days. We've made over 130 investments in small virtual biotechs. So in the ecosystem, I won't prolong my answer, but in the ecosystem, I think it's recognized. I mean, we've been doing what we call venture philanthropy for 20 years. We've been, again, I say this humbly, a leader in venture philanthropy. We were one of the first in this space to create the model. Now you see across foundations, in disease foundations like the Juvenile Diabetes Research Foundation, the Michael J. Fox Foundation for Parkinson's, you see an adoption of this venture philanthropy model where the philanthropies have gone from funding only basic research to recognizing their role in doing what their mission is, which is to cure disease. And the only way you cure disease is by developing a drug, basically. So this has become a more frequent model. Chuck?
In the regeneration of neurons, we know that physical activity regenerates neurons every time you go and exercise. And the World Health Organization has come out with the risk reduction guidelines for cognitive decline and dementia. Can you comment on some of your research? Have you correlated the physical exercise? Is there a way of kind of combining the pill with the physical exercise? In other words, prevention and the pill.
Frank?
We're working on.
Yeah, you're asking a great question about physical activity, and yeah, I think the data is really impressive with physical activity counteracting many of these degenerative mechanisms. We don't know entirely how it works, but one mechanism that's been discovered with exercise is, I mentioned these 2 receptors on the nerve cells, and one receptor is targeted by this very powerful protein called brain-derived neurotrophic factor, BDNF for short. This is a protein that's probably one of the most potent entities known for maintaining that synaptic connection or even regenerating synaptic connections. That protein goes down as we age, but interestingly, when exercise seems to stimulate its production. So for us, that's a tie-in. because one of the groups of drugs we're developing stimulates the same mechanisms that protein stimulates. So in a superficial sense, we could call one of our drugs exercise in a bottle. Now, I don't like to say that because I can't get my patients to exercise. So there are tie-in mechanisms. I think exercise is powerful. It's triggering some of these mechanisms relevant to degeneration, and I think we can harness some of these for therapeutics, and in our case, we have that overlap.
Yeah.
So I think you bring up a very important point that is broader, and exercise is a full body therapeutic. So it's typically very beneficial. The larger issue that you bring up, which is critical, is combination therapies, right? And what's the right combination for the right person and for that right stage of the disease? And that's an emerging area, but again, I think it's an area that ADDF is leading in. And so we've given some thought about what's the right combination of our therapeutic, allo, and a nutritional intervention to support regeneration. And so we're just about starting that actually in the lab. But it's an area that is ripe for innovation and could move very rapidly, because many of these interventions, as you pointed out, exercise, is not an FDA-approved drug. It's an FDA-encouraged activity.
Yeah, I just want to point out from our point of view, first of all, we first heard about exercise and cognitive decline back around, I'd say, 2002. And what we did was we wanted to know the mechanism. So there's a lot of epidemiological evidence. Epidemiological evidence is observational. It only shows associations. I like to give the example of an epidemiologic study that proved that bread is bad for you. And the way it proved that was that it did a study of felonies, and it found that 95% of all felonies are committed within 24 hours of eating bread. So the point is that when we look at these risk factors, we discover them primarily from those kinds of epidemiological studies, but not from randomized trials. So when we talk about smoking or diet or exercise, there's very few trials. Now, recently there's been some trials of what we call multi-domain interventions that show that you can delay the onset. But the question for us is, what can we learn from these lifestyle interventions about how they work that can teach us something about developing a drug? And not just because people will take a pill instead of it, but because there are insights here for developing a drug. So when we did the first study looking at how exercise works, we did a very simple study where we brought 50, 75-year-olds into the University of Indiana exercise physiology program, and they stretched and they socialized and they went home 3 times a week. And then another group stretched and socialized and then went for a brisk 45-minute walk. These were 75-year-old people. We measured brain imaging and volumes over a period of 6 months, and memory and other neuropsychological testing. The people that didn't take that walk, their brains shrunk. The hippocampus, where the disease starts, shrunk according to what we consider normal aging. Their processing speed and their memory function went down according to what we consider normal aging. The people who did that brisk 45-minute walk 3 times a week, a randomized trial, their hippocampus got bigger and their neuropsychological function improved. As Frank mentioned, the next follow-on study that was funded by the NIH looked at BDNF levels in those people and VO2 max, which is what Lance Armstrong would look at when he's on the Tour de France to see if what his fitness is, or any of those bikers. And it showed a direct correlation between exercise, VO2 max, and BDNF levels in the blood and cognition, okay? So it looks like BDNF might be the mediator. You know, if it's red wine, how does red wine work? If it's diet, how does diet work? Is it the fruit and vegetables that are the antioxidants? What are the antioxidants that could be druggable? And on and on and on. So I think the risk, so let me stop there. Okay, next.
For Dr. Watterson, can you tell us a little more about how your drug works, your drug candidate, and also more generally for the panel? Can somebody comment on why aducanumab and all the other amyloid drugs fail?
Good morning.
Okay, I'll let Howard and Frank answer the second question. But for the particular one that Neurokine is developing, That works on a particular enzyme. It's called protein kinase. It's a stress kinase. So it's basically the activity fluctuates during homeostasis, normal day, but as you age, the basal activity of that, as measured by things downstream, things that it modifies, that goes up. And in pathology samples, deceased patients and Alzheimer's studies, the activity that enzyme brings about is also increased. It's also increased in other diseases, and so genetic studies and a combination of basic science studies combined with those observations suggest it's involved in the progression of the disease. So our drug, we actually, while we did the laser-guided design aspect and we used structures that have been used in previous drugs, we did parallel safety studies and metabolism and showed to get into the brain. So we designed it to get high brain penetrance where most drugs fail. I think only 2% of drugs actually get sufficient penetration into the brain to get to the target. So we also did preclinical studies with animal models in which we took the atoms that interact with a drug on the enzyme, and a mouse has a normal enzyme, but key atoms that are involved in the interaction were changed so the drug couldn't bind. When we give the drug to the animals, it doesn't work.
But it's an anti-inflammatory.
That drug, yeah, the drug originally was discovered in industry as being a target for their anti-inflammatory, for their antibiotic drugs, which bring about what's called cytokines, the inflammatory protein going up. So yes, it was discovered as an anti-inflammatory for infection. Later, pharmacologists found it was involved in what is called sterile inflammation, which is inflammation in the brain, inflammation in other parts of the body.
Does it modulate activity of microglia?
It's actually in microglia. It's in several cells. It goes up in microglia. In fact, very stimuli in microglia. It's widely activated, and the drug in cell culture methods it's dose-dependent modulation of the production of these inflammatory modulators. The drug works there, so as well as in vivo.
Yeah, for the technicians in the audience or the scientists, it's a P38 MAP kinase is a target. And P38 MAP kinase, if I'm right, is really in what we call a node.
Yes.
In the pathways of inflammation in the microglia. So successful drugs often affect nodes that have multiple downstream paths effects. And that's basically sort of the technical basis. The key to Marty's work, as Marty mentioned, is that P38 MAP kinase has been a target in industry. And industry is tried to do the chemistry to develop drugs, and they failed. And the key to success with Marty, who's an amazing one of these wizard medicinal chemists, is in the chemistry.
Yeah. So the proof of the pudding is in the chemistry in this case. But also the enzyme is in the neuron also. So what excites us, and why we think it's going to be a very powerful, safe pleiotropic drug, in other words, has multiple functions, is in disease progression, the activity is going up on the synapse and the neuron and these synapse connections, which Frank talked about, and you're also getting this neuroinflammation, and they feed back. We call it the neuroinflammatory synaptic cycle or axis because they're part of an ecosystem. And so you're actually using the same molecular target, the same drug of high safety going in and working there. So at different times during pathology progression, it could be you'll predominantly be affecting neuroinflammation, but you also might be stabilizing or enhancing synaptic effects. And our animal models in another disease actually shows that works. But the point that Frank was bringing up, it totally surprised us. We had a paper in PNAS in December, and this other animal model of disease mimicked on human disease, it reversed the disease, and that was mainly a synaptic effect.
All right, I'm going to ask Jeff to answer the second question about why did anakinumab, which was the Biogen drug that we were all very hopeful for, it really seemed to work, failed. So Jeff, if you want to.
Yeah, sure. Thanks for that question. I think, it's like the hardest question that we're struggling with right now. So I'll give you my perspective on it. One of the frustrations of clinical trials that we just have to acknowledge.
Jeff, can I just back up for a second? So just to explain to the audience for a second. So the question is about a drug called aducanumab that was being developed by Biogen, and it was an anti-amyloid therapy. And even though we'd had a lot of anti-amyloid failures, there was a lot of excitement about this particular anti-amyloid therapy.
Great. Thank you. So when we do a trial and we come to the end, we know whether or not that drug worked at that dose given to that group of patients for that period of time using these measures. That's a very narrow piece of information. And that's all you can learn from a clinical trial. So we don't actually know in many cases whether the drug works if it were given in another population for a longer period of time in a higher dose. So that begins the process of working out those questions. But I'll make one final observation about a drug called BAN2401, which is the current leading compound as an anti-amyloid monoclonal antibody. That drug, in a well-conducted phase 2 study, produced removal of amyloid and a 30% reduction in the rate of decline, which we consider really great. That means that 70% of the decline was probably not attributable to amyloid, which is why all this work is so important. So even if amyloid works, it's not going to be enough. And BAN2401 is a wonderful example of that.
And let me just say, add one more thing. Aducanumab worked. It was a successful trial. Aducanumab worked because it did what it was supposed to do. I think it was 12 or 18 months. Aducanumab removed the plaques from the brain based on the brain scan that ADDF helped to develop to see the plaques. So at the beginning, it was the first trial where all the patients who went into that trial actually had plaques, because all the previous trials, it turned out 35% of the people that went into the trials didn't have plaques, even though it was an anti-amyloid plaque antibody. A Biogen study was well designed, and as I said earlier in the beginning, we learn from everything we do. We're about 50 years behind in developing clinical trials for Alzheimer's disease from cancer and diabetes and hypertension. So we're still on the learning curve. But that Biogen study was a really successful trial because it was well designed, it hit its endpoints, it did what it was supposed to do. 100% of the people had plaques at entry, and then the same test as a biomarker was used to monitor the effectiveness of therapy. And the drug showed on the brain scan that the plaques were gone. So the drug worked. So that's why it was a successful trial, because it successfully and rigorously asked the question, if you remove amyloid from the brain, do people get better? That's the amyloid hypothesis. And the answer to the question, at least from the aducanumab and all the other trials that have been done with amyloid, is no. So let me say there's 150 different kinds of amyloid doses in human beings. And about more than half of them are what we call secondary amyloidoses that are secondary to other chronic diseases involved in inflammation. And they're just like scar tissue. You have a cut on your hand, you develop a scar. Imagine if you tried to treat the scar and not the cut, okay? That's the best I can say. That's the other possibility of what these plaques are. They're just the scars of a degenerative process, and removing them is not going to do anything. Anybody have any other questions? Yes, sir.
Can you comment on sleep and how sleep affects or triggers potentially the progression of Alzheimer's?
Makayla, you want to answer that? Makayla?
Oh, well, there's a lot
Sorry.
of interest in that.
Yeah.
And part of the perspective that
Do you want to repeat the question, please?
Oh, about commenting on sleep in Alzheimer's disease. And there is a lot of interest in that tied to the area that we investigate, because this condition of activity in the brain cycles over sleep-wake. It's not a circadian rhythm. It really is a behavioral rhythm, whether you are awake or whether you're sleeping. And the activity during the day in the brain that is normally higher, and this is the group at WashU, have shown that that is induction, that is inducing of pathology of Alzheimer's disease. And so there is a mechanism where during the progression of Alzheimer's disease, as the pathology is accumulating and this condition of regulating brain activity is failing, sleep-wake cycles get disrupted. So during the dementia course of the phase in particular, there's a lot of difficulty in maintaining the normal sleep-wake function over the day-night period. And there's some indication that in these transitional conditions that becomes more disturbed as well.
Jeff, did you want to comment?
Yeah, I just wanted to comment because I think that's such an important area. And sleep is disturbed in many Alzheimer's patients. So there's this whole part of the pathophysiology, and then there's the clinical problem of insomnia. Merck conducted a positive trial of insomnia and Alzheimer's disease with suvorexant, which I think will change the labeling of that drug at the end of this year. And I think it shows us that we are having some success. It's small, but it's really important to think about these ways of better managing patients. So I'm really excited about the suvorexant trial.
And sleep is a devastating symptom because if you're a caregiver, let's say you're 85 and your spouse has Alzheimer's and you're caring for them in a one-bedroom apartment and a fourth-floor walk-up on the Upper East Side, and your loved one doesn't sleep. And so you're up all night. And it's the sleep disorders that occur very commonly in Alzheimer's are a major risk factor for nursing home placement. Any other questions? Yes, sir.
You touched on it a little bit with regard to diet, but I wanted to ask more directly as I've been reading anything I can get my hands on as it relates to Alzheimer's lately. And a lot of the things I keep coming back to are sugars and removing sugars from the diet, and obviously eating vegetables, eating protein in addition to the exercise as well. Wondering if you could expand on that or thoughts on that.
Yeah, I'll take that real briefly. As I mentioned earlier, the studies on prevention that are going on, which are randomized clinical trials, use what are called a multi-domain intervention. So that means that it's probably that, if you just do one thing like exercise or, sleep properly or don't smoke or don't drink too much alcohol, don't drink any alcohol, manage your diabetes, manage your hypertension, manage your atherosclerosis or your heart disease, you would sort of have to do all of that to really have an impact on preventing Alzheimer's. Diet seems to be important, but again, it's epidemiological evidence. There's never been a randomized trial of diet alone for Alzheimer's. Now, there are some supplements that are involved, and we could talk about that, like fish oil supplements. Maybe not good evidence. So it's a complicated question, but I think my sense of the literature is that it's really, there was a recent study about this, looking at the components of diet, it really looks like it's probably the polyphenols, the antioxidants in the dark colored fruit and vegetables like blueberries and so on, that may be the key component, as well as not eating meat and some of the devastating things. We have time for one more question, Pamela.
So Howard, could you quickly mention the ADDF site that people can go to on?
Oh, thank you. Yeah, so thank you very much. Yeah. So,
I love that site.
That's it, Robbie makes it. So we have a website called cognitivevitality.org. It's Cognitive Vitality, all one word, where we evaluate all of these compounds. If you're looking at supplements, if you're looking at alternative medicines, we have a program. We have 3 PhDs in the office that are funded by an anonymous donor that are working every day to write reports on answering questions about prevention and what's the evidence. So we're really very evidence-based on taking supplements for Alzheimer's disease. And again, it's Cognitive Vitality, all one word, .org. Last question, Pamela, you had a question?
Thank you. We appreciate these excellent presentations. My question is along the line of these recent questions. Has there been a study of teetotalers in comparison to the rest of the population?
So teetotalers are people who drink a lot of alcohol, I guess, or don't drink. Yeah.
So, yeah.
Okay.
Yes.
So I'll take that real quickly. There is an entity called alcoholic dementia. Alcohol is a neurotoxin, and there is evidence that people that drink, let's say, more than 2 drinks a day, have a higher risk of dementia. And in the reverse, there's some evidence, but it's epidemiological evidence. It's like the bread evidence that if you if you just drink 1 or 2 for men, if you drink up to 2 glasses a day, for women, if you drink 1, you have a lower risk of Alzheimer's. Translating that into clinical practice, when I see somebody come into my office that's complaining of memory problems or already has some dementia, I tell them to stop drinking. Because what's the point? I mean, if it's a quality of life issue and you have to have that red wine at dinner, because you've been doing that for 50 years, okay, you can have a little bit of red wine or a little bit of white wine. But basically, on a clinical practice point of view, when I'm making recommendations to patients and I'm trying to optimize their brain health and they're already suffering a bit, I tell them to stop drinking. And the other thing is that we lose brain reserve with aging. So, I have patients that tell me, well, I've been drinking, 4 glasses of wine, and I know the glasses are this big. I've been drinking 4 glasses of wine a night every night since I was 20 years old. Well, when you were 20 years old, you could tolerate it, but now you're 85, and your brain doesn't tolerate alcohol the same way it used to. So on a practice, from a practice point of view, on a one-to-one, I ask my patients to stop drinking. Well, I want to thank everybody. I want to thank our panel. We're going to go have lunch. Thank you very much.
Thank you.